Introduction to hendra virus dynamics in Australian fruit bats, written by Hume Field and Associates, 2011. Hendra virus is a zoonotic agent that is highly lethal to humans and horses. It brings on a set of acute illnesses that often result in death. Known fatalities in horses is 75%, and in humans is an alarming 60%. This brings scientists to want to know more about the virus's host and the dynamics and prevalence that the virus has across all 4 species of Australian fruit bats. This information is vital for the government to implicate a spillover risk management plan. Hume Field and his associates undertook a 3-year longitudinal study whereby they collected pooled urine samples for the detection of hendra virus. The field methods used were spread across 3 geographically different locations that were 1,500 kilometres apart. These were southeast Queensland, far north Queensland and the Northern Territory. Within these locations, there were 6 sites each that were visited every 4 to 8 weeks over a 3-year period. The methods used were 3 by 2 metre plastic sheeting placed under roost sites of an evening, and the pooled urine samples were collected the next morning. They were then treated with a lysis buffer to inactivate the virus particles and preserve the RNA, before being refrigerated and then shipped on dry ice to testing laboratories. These laboratories were the CSIRO and the Australian Animal Health Laboratory. Of the results, 1,672 samples were collected. 311 were in the first year, 465 in the second year, and 896 in the third year. There were a total of 67 sampling events, and of these, 26% detected hendra virus. There was, in southeast Queensland, 26%, in far north Queensland, 41.7%, and in the Northern Territory, 0%. The sites that were chosen had multiple roosts of species. So the sampling events of 2 periods, one period in 2009 where it was an outbreak of the virus and showed an alarming 33.3% of hendra virus detected, as opposed to 2011 where the virus was just simmering. This could explain a certain herd immunity among different colonies of bats. Also, hendra virus was more prevalent in Queensland than it was in the Northern Territory, posing questions about whether location, species and population structure have an effect on the virus. Interestingly enough, the hendra virus detections were higher in the second year than they were in the first and third year, again posing questions as to whether ecological, physiological and other factors may in fact contribute to moderating the infection or to the excretion within flying foxes. To conclude, this study shows that hendra virus is not always prevalent in all colonies of fruit bats, and that excretion can fluctuate over a period of time. Although it is essential to have minimisation strategies, further research needs to be done before a risk management plan can be developed.